Our next speaker today, Dr. Lyndsey DuBose. Dr. DuBose got her PhD in human physiology from the University of Iowa, and we were lucky enough to persuade her to come to the University of Colorado in January of 2020, where she's been working primarily with Dr. Carrie Moreau. And she's going to talk to us today about vascular contributions to brain aging in women. So welcome.
Sorry, technical difficulties here. You'd think for a millennial at this point, I'd be better at this, but. So thank you so much for everyone being here. Happy New Year's. I hope everyone is well. Carrie, are you able to see my slides and hear me?
Yes.
Awesome.
That's good. Thanks.
Perfect. All right, so today, as part of a work in progress, I'm going to present a study that we recently got funding to do. I'll provide a little bit of the rationale in the study design for how the study is going to work, but unfortunately I won't have data since it just recently began. So here are our learning objectives. I'm first going to start a little bit with a generalized background about what we know about vascular contributions to brain aging and the development of Alzheimer's disease. And this will be initially just from the perspective of generalized aging. So my background during my dissertation was really interested in vascular contributions or how the development of vascular dysfunction may contribute to or exacerbate the development of neuropathology associated with Alzheimer's disease. And so a lot of my work was rooted in a study that was published in 2016 during my dissertation work from the Alzheimer's Disease Neuroimaging Initiative, or ADNI. And what this study did was they recruited about 8,000 individuals. They measured brain biomarkers using neuroimaging associated with Alzheimer's disease. And their interest was modeling the trajectory or the probability that that biomarker became abnormal across the cognitive spectrum. So here on the x-axis, you see healthy cognition, early mild cognitive impairment, late mild cognitive impairment, and late onset Alzheimer's. They wanted to understand which biomarker actually became abnormal first during that transition from healthy cognition into early MCI. And what this study demonstrated is that there is a vascular, or the vascular component actually became dysfunctional prior to traditionally studied AD biomarkers, including brain metabolism and amyloid beta deposition. And they measured this vascular component using ASL MRI as a measure of resting cerebral blood flow. And so, these data provided context in addition to the fact that, for 30 years, we've been studying Alzheimer's disease and trying to develop treatments that either prevent the development of Alzheimer's disease or delay the progression of Alzheimer's disease. And unfortunately, those have been largely unsuccessful. A major theme of my doctoral work in my dissertation, my PhD lab, was really looking at changes in extracerebral vascular function. And so a primary theme of that work was really looking at changes in the aorta and the common carotid, which can comprise the large elastic arteries because they have a highly elastic component, in their association with changes in cognition. And for almost 2 decades now, there's been work looking at the association between increased arterial stiffness, measured primarily through the function of the aorta, in changes in cognition. And what we can now appreciate after almost 2 decades is that increases in aortic stiffness are associated with declines in cognition that are primarily associated with subcortical regions of the brain. And these include memory, executive function, and processing speed. Additionally, there's been literature that has shown that changes in aortic stiffness are also predictive of longitudinal changes in cognition, primarily in these domains previously mentioned. But in the last several years, there's been work that's really come out that not only is aortic stiffness associated with age-related cognitive decline, but we now have literature linking increases in aortic stiffness with the development of the intermediate stage called mild cognitive impairment, as well as being predictive of those who are going to transition from mild cognitive impairment to AD. And so these data collectively underscore that changes in the extracerebral vascular function can contribute to the development of neuropathology and cognitive decline in humans. So I probably don't need to tell this group, but the large elastic arteries have 2 functions. So they're comprised of the aorta and the common carotids. And they have a highly elastic layer in the vessel wall. And what this does is when the heart contracts, it generates a pressure that's propagated out into the aorta and the carotids. And that elastic layer within the vessel wall acts to buffer that pulsatile pressure that's generated during cardiac contraction. The second function is that the intrinsic properties of that vessel wall are going to act through Wing-Kessel recoil mechanisms to then conduct steady non-pulsatile blood flow to the target end organs, such as the brain. However, with aging and increased cardiovascular risk factor burden, we know that there are changes in the vascular endothelium that are associated with endothelial dysfunction, and this can contribute over large periods of time to a reduction in the elastic portion of the wall and increased collagen deposition. And so what that happens then is that the aorta becomes stiffer. And so now when that pulsatile pressure is generated during cardiac contraction, it's no longer being buffered, and it's increasing the transmission of pressure pulsatility and pulsatile flow. There's literature saying that the increased transmission of pressure pulsatility is particularly detrimental for the brain, because ex vivo work demonstrates that the transmission of pressure pulsatility into the brain is actually not autoregulated, because it's transmitted at a frequency that is not able to be autoregulated by the cerebral vessels. And so what happens is that becomes a very potent stimulus for cerebrovascular remodeling, rarefaction, and the development of endothelial dysfunction. And so endothelial dysfunction in the brain is detrimental in two ways. First, it uncouples cerebral blood flow supply from neuronal demand, because endothelial dysfunction is an important contributor. Is an important contributor to cerebral blood flow regulation. And second, the vascular endothelium is an important way for regulating beta amyloid in the vascular or the perivascular space. And so when the vascular endothelium becomes dysfunctional, beta amyloid in the perivascular space is no longer cleared. And so there's an accumulation of that beta amyloid that induces blood-brain barrier dysfunction associated with cerebral amyloid angiopathy. And so, while the interplay between beta amyloid and cerebral amyloid angiopathy and neuritic plaques that are characteristic of AD is not completely understood, according to more recent hypothesis of AD, according to more recent hypothesis of AD called the two-hit hypothesis of Alzheimer's disease, it's actually that blood-brain barrier dysfunction that occurs first and contributes to the development of neuritic plaques associated with AD. And so that's a very brief background about summarizing about 2 decades worth of work, but what it provides evidence is that the development of extracerebral vascular dysfunction increases the transmission of pulsatile pressure that has been associated with cognitive declines in the development of AD neuropathology. So hopefully I've just provided some evidence or helped to persuade you that changes in the extracerebral and intracerebral vascular function are associated with brain aging and the development of Alzheimer's disease neuropathology. So, while my dissertation was really interested in generalized aging, primarily from the perspective of healthy cognitive aging, my primary interest is really in women's health. And that's because there's evidence to suggest that women are disproportionately affected by Alzheimer's disease and actually represent about 2/3 of all individuals living with Alzheimer's disease. And while those data are controversial and really are dependent on what population's being studied, in order to try to understand why women may be more likely to develop Alzheimer's disease, what it has allowed us to understand is that women and men do not experience cognitive decline or the development of neuropathology similarly. And so women actually early in the lifespan are protected from cognitive decline, in part because they start higher or perform better on various cognitive performance measures. And so, and this includes memory. So the joke I always like to make is, women never forget anything, and part of that's not their fault because they happen to have a sex difference or a sex preference in memory performance. But what we now know is that while women start higher and they're cognitively intact for longer, they're more likely to transition from healthy cognition to full-blown AD, superseding or passing through that mild cognitive impairment state. and that of women who do develop mild cognitive impairment, they actually decline more rapidly compared with men to AD. One sex-specific risk factor that's commonly been studied to try to understand why women lose this protection somewhere around midlife, of course, is menopause. And so there is one study that I'm aware of that looks at changes in biomarker, AD biomarkers through the menopause transition. And what that study showed is that after a 3-year follow-up, changes in AD biomarkers, there were no change in men in AD biomarkers. However, there were detrimental changes in brain metabolism and beta amyloid across the menopause transition in women. So relevant to the vascular contribution, we have some data from my dissertation that we published looking at mean values, sex differences, and age-related changes in blood flow. But if we look at these data, just looking at their individual scatter plot, and these data are unpublished, so looking at changes in blood flow across the lifespan with women shown in red and men shown in blue, what we find and what's been demonstrated in the literature is that women have higher cerebral blood flow early on in life. However, in late life, this female advantage is lost. And more importantly, if we look at that point in which they intersect with each other, it seems to occur around midlife and around that time that a woman's going through menopause or shortly after. And so this really led me to be interested in what are the vascular contributions to brain aging and how do these vascular changes or vascular functions change with menopausal status at midlife. So cross-sectional data looking at changes across the lifespan or in different age groups in women show that, while there are sex differences in in early life with aortic stiffness, women have an increased change in or have a greater change in aortic stiffness across the lifespan. Similarly, work by my mentors, Carrie Moreau and Carrie Hildreth, they've looked at midlife and have shown that increased carotid artery stiffness, which is another large artery, increases progressively across the menopause transition. And so while these are cross-sectional data, they show that menopause may be a sex-specific risk factor for an acceleration in vascular aging that might be important for studying within the context of the brain. So in order to try to look at sex differences in this vascular contribution, these are cross-sectional data that we provided as preliminary evidence. But what we did was we looked at sex differences in the association between aortic stiffness and basal cerebral blood flow in the hippocampus. And what we found was that increases in aortic stiffness were associated with lower basal hippocampal cerebral blood flow in women, but not in men. And so we took this a step further and we wanted to look at the association between hippocampal cerebral blood flow and performance on a delayed memory task that of a previously learned word list. And so what we also find that is in women, those who had higher cerebral blood flow also performed better on delayed recall. And so we use this as preliminary evidence to say that maybe women are more sensitive to changes in the vasculature and that they might have an increased vascular contribution to brain aging. Now, the limitations in this study, of course, is that these are cross-sectional, and as well as all women are older and postmenopausal and had been postmenopausal for about 5 to 10 years. And so it really limits our ability to look at underlying mechanisms associated with these sex differences that were observed. So as a second summary, the menopausal transition associated with detrimental changes in regional AD biomarkers, and that there's cross-sectional evidence that suggests that perhaps there's an increased vascular contribution to brain aging in women. However, to date, there hasn't been any studies that have looked at this. So a primary limitation in the data that I presented previously, of course, is that when you study menopause, it gets a little bit challenging. One is that commonly when we look at menopausal groups, these groups differ by age. And second, if you look at longitudinal studies of menopause, as a woman goes through menopause, they're also getting older. And during that time, they're also accumulating an increased cardiovascular risk factor burden. And combined, these two can confound some of our ability to really isolate the effects of menopause. Now, one way that we can try to isolate the effects of menopause and reproductive status is to use a pharmacological ovarian suppression model. And so ovarian suppression models have been endorsed as an appropriate strategy for investigating the regulatory actions of estrogens and sex hormones in women. The way we do that is we administer a gonadotropin releasing hormone, either an agonist or an antagonist. And for the perspectives of this following few slides, I'm going to talk about antagonists. And what these do, these temporarily and reversibly lower endogenous sex hormones, all endogenous sex hormones for weeks to months, depending on the drug and the dosage. So GnRH antagonists act on the anterior pituitary to lower LH and FSH release. And these can, but aren't always coupled with estrogen add-back or placebo add-back. And what that allows you to do is to isolate the effects of the individual hormone being added. So in work that was recently funded by the NIH through an NSA fellowship, as well as the Ludeman Center, to try to understand the roles of menopause on vascular function or the vascular contribution to brain aging, and to understand how changes in individual sex hormones contribute to those observed changes with menopause. And so the way we're going to look at this is we're going to recruit pre- and postmenopausal women between the ages of 45 and 55 to allow us to age match women. All postmenopausal women will have gone through natural menopause, and they'll be overtly healthy without any presence of disease, and they'll be cognitively intact. So the way we're going to look at the influence of menopausal status on vascular function is two different ways. So first, we're going to look cross-sectionally by recruiting pre- and postmenopausal women, and we'll study vascular function at baseline. And so our primary outcomes will be measuring aortic stiffness and pressure pulsatility, because again, we think that may be an initial, that may cause downstream changes in the brain that are associated with AD biomarkers over time. We'll also look at the intracerebral vascular function, and we characterize this by measuring regional or microvascular cerebral blood flow using ASL MRI. These data are in collaboration with Andy Callan in the Department of Radiology, as well as other indices of cerebral blood flow regulation using transcranial Doppler and changes in cognition. Now, the limitation in studying menopausal groups is even if we age match, there are still differences in multiple sex hormones. And so this really limits our ability to isolate the contribution of an individual sex hormone. So of course, in premenopausal women, as everybody knows, estrogen's high and FSH is low, and the converse is true in postmenopausal women. So the way we'll overcome this is that we are going to enroll the premenopausal women into a 12-week GnRH antagonist intervention. And so vascular function will be assessed after a single injection of degarelix, and that injection lasts for about 12 weeks. And what we'll do from there is we're going to look at within group changes in vascular function within those premenopausal women with following the GnRH intervention. And we'll also look at the values post-intervention in those premenopausal women, how they compare to the postmenopausal group. So your third summary here is that while studying menopause is challenging because of changes in age and risk factor burden during the menopause transition, ovarian suppression models can be used in order to isolate the roles or isolate the effects of chronological aging from reproductive aging. Now, I've been giving this talk for a couple of years, and I've been talking about my work for a couple of years. And a comment that I commonly get is that, talking about menopause and AD, it feels a little doomsday, and that, all women go through menopause, but only a small percentage develop mild cognitive impairment in AD is a comment that I typically get. And usually what I respond is, yes, and I agree completely, and I couldn't agree more. But just because a woman doesn't have changes in cognition that are noticeable, it doesn't mean that they're not developing neuropathologies that has been associated with changes during the menopausal transition. And so if we're going to ever make a change or have a positive impact on sex differences in AD, we have to really understand what the sex differences that are contributing to the development of neuropathology, and if this corresponds with changes in menopausal status. And what I think it's important to note is, while we don't have treatments to prevent AD, we do know that the female advantage seems to be lost somewhere around midlife, and that AD is actually increased in women that go through menopause earlier or go through surgical menopause. And so, really what I think this work will do is it provides the baseline and the foundation that we can then look through we can look at the intersections of menopausal status with other risk factors. And so, there's many people here at the university that are looking at many of these different factors that accumulate across the lifespan, both in terms of cardiovascular disease risk and menopausal or cardiovascular and Alzheimer's disease risk. And so my interest is really looking at I agree. I don't think it's just menopause, but I think it's probably something related to a cumulative risk factor burden that a woman might, or a man, might accumulate these risk factors at different periods of their life, and that intersects with changes in their reproductive status or their gonadal hormones that may actually exacerbate through an accumulation of these risk factors. And so my hope is this helps us to understand what happens to the brain during menopause so that future studies can look at individuals with different risk factors accumulated at different time spans. So I've only presented a very small portion of the work that's being done on this study. And I'm fortunate enough to be mentored by an entire team of individuals, both here at CU, who have been fantastically supportive throughout the 2 years and during COVID delays, as well as individuals at other universities. I am a member of the IMAGE group, and it's a hugely collaborative and wonderful group. And so I'm very grateful. And these works are not possible without their input, as well as our funding sources. So I'm happy to take questions. This is a shameless plug. We are recruiting. We are recruiting both for the study that I just presented on ovarian hormone control of vascular function, as well as 2 additional studies looking at different aspects of pregnancy and vascular function. And so I'm happy to take questions. And if you have any comments or are interested in these studies, please email me.
Thank you, Lindsay. That was great. Really appreciate that overview of your work. One question has come in, and that's whether changes in aortic and carotid stiffness always track with age?
There is a heavy age component to aortic stiffness. I'm trying to think if I think in adolescence, it doesn't necessarily track with aging, in part because of changes in body composition and obesity and growth. And so you don't see a similar relation between age and aortic stiffness in young adults. However, it is nonlinear once you get into adult lifespans. And that, I think, is where there's a sex differences in that age generally has a linear association with aortic stiffness in men, but not in women.
Thank you. Another question, how do estrogen replacement and statins affect aortic stiffness?
Great question. So talking about the first with hormone therapy. So I'm trying to think, and I think you guys have done some of these studies, so I'm going to blank on some of your study results, but from the perspectives of estrogen replacement, I think it has been shown to improve large artery stiffness, but I think that's dependent on when it's being administered. In terms of statins, I'm not sure. I think those data are relatively mixed, with some studies demonstrating a benefit of statins and others not.
Thanks. And I don't know if Dr. Moreau wants to weigh in on that as well. She may have some additional things to say about that.
Correct me.
Another question is, in your study following the premenopausal women longitudinally, will you also collect subjective measures of cognitive function, like how women are experiencing their cognitive functioning, so their perceptions of how they're doing?
Yeah, that's a great question. So we do have a survey that's getting at symptoms with hormone suppression. And so I think within that, we're also looking at, like, do you experience brain fog? Do you feel that you've had changes in cognition? So we will be able to look at that. I think there's some literature out there that subjective cognitive declines do increase with the menopausal transition, but these aren't necessarily associated with the development of mild cognitive impairment. That being said, if a woman feels that they're having changes in their cognitive state, of course, that's certainly very important to note.
Thank you. And just a response from Dr. Moreau regarding the question about estrogen. So she says estrogen treatment has been shown to improve stiffness, arterial stiffness, but the effect may lessen depending on age and as well as the type of treatment. So for example, whether it's oral estrogen versus transdermal, and statins have been shown to improve arterial stiffness, but it's kind of, the data are mixed on that.
Cool. Thanks, Carrie.
Thanks, Carrie. Another question, is having had COVID a possible covariate given the emerging evidence regarding the impact of that infection on cognition?
Yeah, I think that's very possible. It's not, it's something we'll collect in somebody's medical history as we do the study, of course, and we'll ask as they go through the study. I know, a number of people are experiencing some of those challenges right now, but I can't necessarily say how that will interact with menopausal status on cognition right now.
Not seeing any more questions in the chat or Q&A. Any final questions or comments from participants? Dr. Moreau just comments a large study out of Europe looking at COVID and arterial stiffness. So it'll be interesting to see what that shows.
Yeah, and hopefully they look at sex differences, but I'm not sure if that's a primary outcome right now.
All right, well, thanks everyone for joining. Thanks to our presenters. It was great work by both of you guys. Will be exciting to follow what you guys do ongoing. So we have a few minutes. Remember, division meeting, and please check your email for a link to an evaluation for today's presentation. And we'll see you in a couple weeks. Thanks, everyone.